Well, welcome back, James.
Thank you, Leighton. I enjoy being back here with you guys.
We enjoy your brain power, my friend. So I think I'd like to start today talking a little bit more about whether, and I know that like there's both anaerobic and aerobic gram-negative and gram-positive bacteria, but are endophytes typically a gram-negative, or does it matter?
That's a good question. You know, it doesn't seem to matter, but we also don't know what all microbes are endophytes. I mean, for example, there's some evidence that we could have, besides bacteria, gram-negatives and gram-positives, but some people have reported archaeans, right? So very primitive group of bacterial-like of prokaryotes, right? And so those have been reported, but more recently, people are finding that there are also protozoans, possibly protozoans. We did a study with this landrace corn, and we found in the landrace corn that using the fatty acid analysis, looking, and you can actually track that back to the source of the fatty acid, and we find that the protozoans are in there. There's some gram-positives. Well, of course, gram-positive bacteria, gram-negative, but also the rhizobial bacteria were indicated to be there, and protozoans. So, and we've also been able to observe that algae can also be absorbed into plant roots, and we've seen that one time in a European sample. I've seen algae in actually root hairs in a screen, and then we did some collaborative work with In-Soil Algae, a company, and we actually looked at their alga, their living alga, and those algae carry bacteria with them. And so when they're absorbed, this chlorella, when they're absorbed into roots, those bacteria can be released into the plant. So it's interesting. The situation looks like a lot of different microbes can actually be endophytic. We don't know what the character is, how the plant determines which microbes to take in, what are the qualities of the bacteria or microbes that come in. It's hard to see a common denominator between them. The only common denominator is that they appear to not be aggressive to the plant. There may be other things, like, for example, do they secrete choline, and is that a signal to the plant to take them in? And that's a possibility. That's one hypothesis that we have, but we really don't know. The rhyme or reason why plants, how plants determine which microbes to take in. We don't know. It's a good question, Leighton.
Wow. All right. So that just opens up so many more, because I mean, from what I understood, there has to be a loss of either the outer shell or a disassociation or degrading of the shell in order for it to enter into the plant cell. So when you get into
actually, they take them in. They take them in with the wall on, and then they remove the wall like that. Then the plant removes the wall. Okay. So yeah, they have their wall to begin with. And you're right; those changes happen. So what is it that causes the plant to do that? We don't really know. We think ethylene is a key signal, and that causes the plant to secrete the reactive oxygen onto the microbe. But it may be, people are doing interesting things. For example, they're taking cell walls of microbes, and they're killing the microbes. They're taking the cell walls, and they're using that as a kind of a fungicide, right, to try to trigger the immune response of the plant. So the plants can detect these microbes, and they may just be able to detect pieces of the microbes, pieces of their cell wall, for example. And I don't know exactly what they're detecting, but there are substances on those walls that they can detect.
So chemosensing of some kind.
Some kind of chemosensing is going on. There are people who look at they call them pathogen molecular patterns, right? Pathogen molecular pattern, something like that, having to do with pathogens, right? And it's basically that the plant can detect what these microbes are putting out, and then have an immune response. But that's some. I don't know a whole lot of details on that.
Yeah, that's next level for sure.
That's next level. That's next level. Yeah. Now that doesn't mean we're not getting in the next level. We're trying to get into the next level, but it takes time to get there. It's we were just kind of peeling in a little bit at a time, and trying to figure out what is the important thing that we can work on, right? What's the next important thing? For example, the question of how microbes go inside plant cells, inside roots. That's an important question. So we keep trying to get to it.
So when you talk about protists that are also endophytic, aren't they a lot bigger than the bacteria or the bacterium?
There are these, but they all have stages that are different. I mean, consider a protist. The protists, some of them at least have soft cell walls, right? Or they're like they can move, right? So, and you always have this. Don't forget, you always have different sizes. So, I mean, this is an issue with the algae, for example, that I was the chlorella. The cells get pretty big. So, how in the heck do they get into the plant cells? What we think is going on is when the algae are dividing, the dividing cells are much smaller. So those smaller cells can be taken in. The larger cells probably not, but the smaller cells, the dividing cells, the actively growing cells, those are the ones that can probably be absorbed.
And then it would beg to question, how are they going to grow? Maybe they're limited to growth based on the actual plant cell walls, so that therefore they can't just like explode the cell wall.
Well, they get hit as soon as they go in. The algae themselves get hit with superoxide.
Oh, so they're turned into food.
So they're turned into food right away. Now we can see their empty cell walls inside, but when they're hit with superoxide, once they're absorbed and they're hit with superoxide, the algae themselves carry their own bacterial endophytes. So that when they're hit with superoxide, those algae leave the bacteria leave the algae, and we can see the algae losing their chlorophyll. So it's it looks like the plant is munching down on these green algae, and getting their endophytes out of them. Okay. Now, how important is that? How significant is that? Well, looking at the corn, they take in lots of different microbes. So they're taking in communities of microbes. That's what it looks like. Communities, maybe lots of different variety being absorbed into roots and then going through roots like that. So it's interesting situation.
Yeah, it is. And chlorella has, just blown up on the scene as the go-to algae for crop production, which has all happened within the last few years, as far as I can tell.
Well, it's really it's easy to grow chlorella, for one thing, because it has a saprotrophic phase, right? It can feed. You can grow it in fermentation cultures in batches, right? So you can't do that with a lot of algae, some organisms, but with the chlorella, they can ferment it, and you can grow a lot, and they can even grow it under conditions where you just feed it sugar, so basically you don't have to have the light even. So it's
Interesting.
possible to, yeah, no, it's more flexible. Chlorella is a more flexible alga. You could go for some of the other Duniella or some of the other algae that people will sometimes grow, but they're not easy to grow. They're much
Well,
harder to grow. Yeah.
that's like the work that's being done down at Santa Fe Community College. They were one of the early on adopters of algae technology. They have everything from I forget the name of it, the one that's grown in salt water. Yeah, it's a superfood.
Is it okay? It's the it's not the spirulina thing, right?
Spirulina, yeah, spirulina.
Okay. Yeah.
And then they also got into a couple of different strains that were really productive in cannabis cultivation, where they sent up to, they had a client up in San Francisco or just North Santa Rosa who was buying totes every couple of weeks and introduced it into the legal cannabis community, and we're having tremendous results with it. And I don't know what that species was, but maybe it was Chlorella.
Could have been chlorella. I think that's probably one of the most popular ones. And it really, when you think about all the things that chlorella, because because it has chlorophyll, so it can photosynthesize. So basically, you have a problem when you put biostimulants down. If if they're, for example, if they're like bacillus or something, you put them down, and it's well and good you put them down, but they go dormant right away. They go into endospores because there's no food there, right? So they form their spores, they go dormant, they just lay there. The difference with something like chlorella is that you put that down and it will continue, provided there's moisture there, it will continue to live in that soil and on that soil and continue to create a kind of a living environment around itself, putting out bacteria too. So not only creating an environment where other bacteria can grow around it, but it will be putting out its own endophytes in the environment. And so plants, can have this kind of ongoing, instead of dormant microbial supply, and a living microbial supply around these algae, right? So it makes a lot of sense as a biostimulant. It has some advantages over some other kinds of just putting straight microbes down.
Interesting. So another question. Maybe you could do a little bit deeper explanation of what you recently discovered, where a bacteria goes inside another bacteria and eventually forms an oregano or forms an oregano and then forms a nucleus when that bacteria divides. What is that process called?
When the bacteria go inside plant nuclei.
No, inside another bacteria.
Oh, inside a bacterium in an endosymbiosis.
Say that again.
Endosymbiosis.
Endosymbiosis. All right, I'm gonna choke on that.
Yes, endosymbiosis, and that is interesting, right? The phenomenon that gave rise to eukaryotic cells. The mitochondria are thought to come from purple sulfur bacteria that were absorbed into the plant, and then you have chloroplasts that came from other blue-green algae that were absorbed into plants. So that phenomenon. So what we're seeing in plants is really a bit deeper than I would say the tip of the iceberg would be the endosymbiotic theory, but actually endosymbionts are everywhere. Plants are full of them, and fungi are full of them, and animals, we probably have them in us too. There are probably bacteria that are in there that are not causing disease. Maybe they cause disease later on. I don't know, but we don't work on animals, so we can't say much about them. But definitely plants are filled with microbes, and so are fungi. They're filled with microbes.
Have you many studies on fungi allowing the endophytic process to happen?
So they're in fungi. So they're in. I have a graduate student working on that, uh, and they are actually. Our overall hypothesis is that fungi and plants are cultivating bacteria in their meristems, right, in their dividing tissues. So that's kind of where the student is working now, Nicholas Jansma, but he's looking at the microbes that fungi cultivate in their growing tissues. Eventually, we want to figure out exactly what these microbes are doing for the fungi, and just like we want to figure out what these microbes are doing for the plant.
Yeah, so I saw a really interesting series of I want to call them loops where they used a specific microscope to actually document mycorrhizae fungi in situ in place, and they showed the bacteria actually, I guess, losing its shell and entering into the fungi and then being transported to another plant. We assume, we don't know, because it's not like you can continue to chase that forever.
Was that a mycorrhizal fungus? Mycorrhizal?
Yeah, yeah, it was a mycorrhizae, and
Mycorrhizae are full of bacteria.
right.
Mycorrhizae, yeah, they're full of bacteria. They really are. They're loaded. They don't have any septa,
Hmm.
so the bacteria, once they get in, they can just go all over the hyphae. So when they take them in, they have a lot of bacteria in them. That people have been studying that for a number of years now.
Yeah, and I just found that like so powerful because they also showed that there's an aqueous coating on the outside of the mycorrhizae, similar to like a rhizosphere, and that was transporting these
It's
organisms.
true. It's true. It's true. And it becomes like a highway, like all hyphae. In fact, if you look at any hyphae in the soil, you think of fungi. Fungi are not there alone, right? Fungi have bacteria with them all over their hyphae, and some go inside the hyphae. So it's a little bit different than we normally think. We think of microbes as discrete, right, or organisms as discrete, but in fact, they're not completely discrete.
No, that's for sure. It's just a really wild world when you start thinking about like
It is, and
right.
sorting it out, figuring it out, it's going to be very hard because later when you think about it, when you think about a bacterium, right, and a plant, for example, or a fungus, you have they're so tiny, right? They're so tiny, and the chemical interaction between them is just in such a tiny micrometers, right? A micrometer, 2 micrometers, and so that interaction. So how do you tell that? Imagine that. How do we tell that? We could look at it, and we could see it, and we could play around with chemicals to put chemicals there to see what might be happening, but it's very hard. I don't know. We don't have the technique yet to get down in there and measure right in there between that bacterium and that plant to measure what those chemical interactions are. Right? We're guessing, we're trying to put everything together based on what knowledge we have and what experiments we can perform that might tell us. But we're on a primitive level right now.
Do we have any hope in the near future with AI?
See, you have more faith in AI than I have. I mean, you have more faith in AI. I I I think, yeah, it'll be a useful tool. It will. It'll help us crunch stuff, right? Crunch data and stuff like that. Yeah. Will it really? I don't know. I've heard other people say, and this is for discussion. Somebody said they're going to worry about AI when AI has legs and starts going around the lab, right? Starts walking around the lab and starts running jails and stuff.
They
That'll
already got, dude. They already got him out here in LA making burgers and fries, so it won't be long.
Really? Oh my gosh.
Yeah, no joke, man, no joke. Um, so, so interestingly enough, when you really go down the rabbit hole of these interactions, and you start to say, okay, well, horizontal gene transfer is crazy enough, but then you move into this next level of where the bacterium is absorbed into the other bacterium, and then they get divide and turns into a new nucleus. Is that considered a new species within the realm?
If the no, what in the corn we're talking about, for example?
Yeah.
Yeah. In terms of genetic changing, right?
Yeah.
Not a new species, no, but it may be what we think it is. Okay, let me go back.
Mm-hmm.
Okay, so because it's kind of crazy, really, when you think about it, it's kind of crazy. But what we see is these microbes growing in dividing tissues, dividing cells, and they're going into the cells that are dividing, right? They're inside these cells that are dividing. They actually will go into the nuclear envelope. And we think what's happening is when the cells divide, the nuclear envelope comes apart, right? And the nuclei, the chromosomes separate like that. Okay. But if there are bacteria in the cells now, they're already in the cells. When that nuclear envelope comes back, reforms, it wraps some of the bacteria up in that nuclear envelope, right?
Yep.
And they get trapped in that nuclear envelope. So they're in there and they're producing chemicals like ethylene, and we think nitrogen too. We think nitric oxide. Well, we think nitric oxide and maybe nitrate, but there's definitely ethylene there because we can detect that. And that ethylene is known to affect cell division. It's known to cause plants nuclei to divide, to become polyploid. So we think what could be happening is that the nucleus, that these bacteria secreting ethylene and maybe nitrogen oxide, other forms of nitrogen, might be causing the DNA of the plant to change. I mean, in the sense that if it's replicating, one thing that we have happening then with the replication is jumping genes. In corn, there are these transposons, right? These genes that move around, transposable elements, they jump around and they move traits with them. That's the point. So they generate variation. Okay, so the idea being if the microbes are there, they might be increasing variation. And that's not important to everyone, but to a breeder who is trying to get new variation, new breeding material that's more, I don't know, more resistant, more hardy for environmental change, whatever it is, whatever they're trying. They may be able to get more variable material with endophytes than without. That's the so endophytes could benefit the breeding program. And that's the whole point of that. It doesn't make a new species, but it makes more flexible, more variable breeding material, right? More offspring, more variation. That's what we're looking at. That's what we're trying to evaluate and determine that's one of the things we're trying to determine if that's happening.
So that would be typical, like, of the offspring. So the seeds that came from that plant.
Yeah, it would be the offspring that show that kind of variability. Well, I wouldn't. I hate to say that only the offspring because there is such a thing as epigenetics, right? And we mentioned epigenetics before. And so having endophytes in the parent plant will turn genes on in the parent plant too. I mean, that's one thing about endophytes is when you get plants with a lot of endophytes, you see a lot of variability in those plants. And that's because some plants have some endophytes and some plants have others. And so they'll respond differently, right? As compared to if you were just to grow plants with chemical fertilizer, then you get plants that are real big and more even, right? So the endophytes may be not only causing variability in the plant, the mother plant, but also causing variability in the offspring.
Do you think it carries through in the pollen as well, or just the
We see. Yeah, you read my mind. We see We see the endophytes in the pollen. We see the endophytes in the pollen. So do those genes, I would expect they do. Certainly endophytes are going. We could look at pollen grains and pollen mother cells. Pollen mother cells and pollen grains are full of bacterial endophytes. So they're definitely there. Now, they may not be the exact same endophyte that you have in the root, right? There may be a different one that's that's mostly working in the shoots and going into the fruits and the pollen, and so the leaves. Okay, so it's still an endophyte, and they're everywhere in the plant, different ones.
So it goes to show you how, in theory, these plants can keep up with environmental impact in a situation of, like, a reset, an earthquake, a volcano, a tornado.
In fact, there is a colleague of mine who, uh, Rusty Rodriguez and Regina Redmond, to a married couple colleague that made a hypothesis, actually, and that hypothesis is let's see what it was a term for it, and I don't think I can remember the term. It's a habitat adapted symbiosis. Habitat adapted symbiosis, and this is the idea that the plant can create a symbiosis with microbes to help it survive in a particular habitat. Like, for example, salt stress, right? Where there's salt in an environment. Well, an endophyte can associate with a fungal. I mean, a plant can associate with a fungal endophyte. That, I mean, we've have shown this happens a lot of times where a plant will associate with the fungal endophyte that provides it with stress tolerant, so it can grow in that high salt environment or whatever the environment is. So the idea basically that plants can survive or thrive in an environment by selecting the right symbioses, right? Habitat adapted symbiosis. That's the idea.
Wow, wow, next level. So, again, all the more reason to stay away from chemical fertilizers and chemistry, because you're really limiting yourself to.
Yeah, that stuff is really bad for us too. So
Yeah.
much of it is. So much of it is.
And of course, it's getting into waterways, into our food,
Yeah.
and everything else. So,
Yeah.
but again, the future of agriculture should be more faced with conversations like this, and not this typical, oh, well, we're making a new biostimulant. We're just adding less urea, right? Hello, come on, people here. Let's get on the same page. Um, so I guess the other question is, and this gets more toward the chemistry side of things. Like, for instance, plants that have taken up a lot of salt or a lot of chloride. Is that the reason that they're not dying? Is because they've got that relationship in them that's preventing that excess chemistry from hurting them or stunting them?
It makes a lot of difference, Leighton. Plants are much hardier when they have all their endophytes, and when you remove those endophytes, they're weak. They're really weak. So yes, I would say likely the ones that can survive in that environment are surviving because they're genetically capable, right? And that's what we would have said without thinking about endophytes, right? Or because they have endophytes that make them resistant, that give them increase their hardiness, or even could even be breaking down toxins or whatever it is. But more than likely, it's an oxidative stress uh, resistance that you're seeing in plants that grow in those real chemical laden environments or stressful environments.
Yeah. So one of my clients, he's a groundskeeper for a national football league, and he has been chemistry free for probably 5 years. There was a shift in management, or a shift in ego, and they brought in a new chemistry orientated, typical greenskeeper to recommend a new regimen of chemistry. And so we've been actually watching the reverse way. Typically, what I'll do is I study, the path that goes forward as we remove chemistry and increase biology. I mean, that's what I do. So this is the first time I've ever been exposed to the reverse, where we had an incredibly biologically diverse, we had mycorrhizae colonization up in the 40s and 50s, which is excellent. We had tremendous amount of saps, as well as the full mix of protozoa. Some ciliates, yes, because of the nature of the draining of the field, that there were areas that were excessively wet. We also had some root feeders, but they were kept at bay by the predatory. So, I mean, we're talking though a very, very biologically diverse system until the new guy came in and started throwing a bunch of chemistry at it. Now, we haven't seen the decline that I expected. And I'm blessed that Chris is continuing to work with me, even though I'm technically really not important at this point because it is chemistry. But the chlorides and the salts in this grass is just through the roof. And it's just a mind scratcher to say, how can these plants tolerate that? So I think you've just answered the question, is because of how biologically diverse we had built that system up to, we probably had a tremendous amount of endophytes, and perhaps even, to a level that they're able to combat what a typical plant that had not all that association could deal with. What are your thoughts on that?
It could be, yeah. No, that makes sense. If you built up a healthy regenerative soil, those soils are going to have resilience, right? There's going to be some resistance there. So even some chemistry may not ruin that.
Interesting. That's what we're kind of witnessing here.
Kind of watch it and see. I mean, it might be valuable to know that you could, bring back soils and then apply a little bit of fertilizer after that.
Yeah, I mean, it appears, and again, it's not just the fertilizer. They're forced to use what's called reclaimed water, and that water is ridiculously nasty.
Oh, really?
Yes. You know what I'm talking about. This is the stuff from wastewater treatment plants, and it has not been fully treated.
Gray water, right? Is that
gray? For lack of better words, it's gray for sure. Huh. And again, when I figured, oh God, I mean, in the beginning when we first started working with him, which was probably like 5 years ago, maybe even a little bit more. We really struggled to get those level of both protozoa, nematodes, and fungi up to speed. But once we got them there, it appeared that the reclaimed water was not, as harmful as it was prior to getting involved with the biology. I mean, from what I remember, he told me that the sod replacement was like a regular routine throughout the course of the year because the water was so contaminated, and that with chemistry just wouldn't allow the plants to survive. So again, this is like a whole new level of resistance that I didn't anticipate.
Yeah, well, we expect it really. I mean, it does make sense, right? That you have a good biological environment there, regenerative soil, and the plants can take more. They're going to be oxidatively resistant, so they're going to be tougher than if you had poor soils.
Yeah, and just to take it to another level so you can visualize this. This field is open on one end and surrounded by 12, 15-story glass office buildings on all three other sides. So when that sun hits those windows,
it's cooking them. It's cooking them down there. It's like Austin. Like when I was in Austin, Texas, there used to be this big gold building. It was in Austin, downtown Austin, the sun in the summertime would reflect off that, and it would be like being an ant. in the sidewalk under with a magnifier, you know.
Are you admitting that you did that when you were young too?
I did that. I did that. Yeah, we all did that, I think. I'm
Yeah.
sorry
So,
I haven't done it.
right? So yeah, we're admitting our age, or I should be showing our age anyway.
Mm-hmm.
Yeah. So again, like, I've just been, blown away by how that increased biology has just supported this, plant health in a condition that is just ridiculous. And again, if he doesn't get rain, and he's forced to use pure recycled water, reclaimed water, um, the salt levels go through the roof. The only time he gets any reprieve whatsoever is when they get a heavy rain that helps to wash that down into the lower profiles. Because he's still, at his best, I think we had 4 to 5 inches of root mass before the root hairs, right, in depth in the coroplugs. Now he's down to about 2, maybe 2.5. Now that he's back on chemistry, which is going to eventually cause a problem because they have what's called a torque test. Imagine a 300-pound muscle-bound linebacker, and if he loses footing, he can rip muscles in his legs. So there's this player health relationship with the turf that the NFL requires that you have a certain level of this torque test to prevent those kinds of athletic injuries. And right now, he's barely passing that test. Whereas before, he was up in the 30s. Now he's down around 10 or 12. Right? 10 is the lowest you can go. And again, so you start to really think about that, compounding that like out because of how much root mass that was going to be providing, that much more biological tolerances within that plant, because again, the root's pulling up what it needs to support what's above the ground. So it's almost like it's going to hit a point and collapse. And, that's going to be an ugly day for a lot of people in that program.
Golf course you're talking about? Golf course?
Yeah, actually, turf. It's a sports field. High
field. What kind of soccer or?
No, football.
Football field.
Yeah. So an NFL, and again, you've got some really. Think about the dynamics of the different players, like from the running backs that are doing this cutting and bobbing and weaving, to the big boys who just stand and dig their cleats in and are pushing forward. Again, either one of these, they lose resistance or they lose traction. That's when you have injuries. So it's a real challenge. And if he goes below that threshold of, I think it's 10, that's a big issue. And then again, we've lost 50% more of our root biomass. So that's eventually going to lead to a weaker plant on the top side. And then as that plant becomes weaker and weaker, I've got to assume the chemistry has got to be affecting the endophytes and the other internal biology.
What they do is lean more and more on the chemistry. So as things fall apart, they're gonna probably apply more chemicals.
Yeah, no, there's no doubt in my mind until they pull the plug and say, okay, now we're back to sodding 3 times a year.
Yeah.
What is the cost of this?
Right. That'll happen. It's a chemical trap. You get into that.
Yes.
One thing turns to another, and it's like you're trying to stimulate the grass so the grass will grow, and as a result of you put your fertilizer on, and then the grass grows and you have a lot of disease. So then you got to put your fungicides on it, right? And then before you know it, you have grubs eating up the roots. Yep. So you got to now put some insecticides on it. Before you know it, you got toxins all over the yard.
Yep, and then the grass gets weak, and
it's weak. More disease, more chemicals, right? And that's how they do it. That's how they do it.
No, I know, and it's just again, this is the first time I've watched it go the other way. I'm very comfortable transitioning dirt into soil, but when you're taking, beautiful regenerative soil and then just degrading it and watching it go to the wind. It's not a fun place to be, especially after 5 years of hard work and getting it to the level that we did. But hey, it is what it is.
People think differently, and they go one way, and I mean, and then it maybe was working just fine, but they think, well, maybe if we did this like everyone else, then we'd get something better, right? So they switch and try to do something better.
Yeah. Unfortunately, the nature of humanity.
It's the nature of humanity, exactly.
Little good, more is better.
Yeah. We go back and forth, different things, trying to get something that works right.
Well, listen, typically we take questions about 2/3 of the way in, and we already
we can do that.
2/3 of the way in. So there were some good questions. Ken, you want to hop on and go ahead and ask the questions?
Yeah, go ahead.
Sure. The first one, or in fact, the first three are from Case Gilmore. In Dr. White's best opinion, what's the best way to feed the plant as a non-microorganism?
So what is he saying? Biostimulant?
To get the plant, so non-microbial way, right? Non-microbial way. I think if you now not feeding the plant microbes, but feeding the microbes, feed the microbes. In other words, cultivate the microbiology rather than the plant. If you do that, I think you'll have an interesting outcome. People do things like, put a little bit of sugar, for example, in the soil to make a jump, to make the plant grow more. And what that does, it makes the microbes grow more. And of course, more nutrients also get released, but more microbes grow so the plant can get more microbes. So thinking about growing the soil microbiology and cultivating soil microbiology as a focus instead of thinking cultivating the plant, right? Cultivate the microbes. And just however you do that, with just a little sugar or some other kind of a meal, it could be soy meal or something like that, or basically it's a microbial focus. Now I hope I didn't ruin that because you said non-microbial, but I assume that you meant some other way than putting microbes on the plant.
I think you're spot on. I think that's what he was trying to ask. And,
okay.
And he can clarify because he's still in the chat.
It's a good question. Good question.
Yeah, and I think you're right though. Again, is you start feeding the soil and not consider the plant.
I think you take care of the soil, and you take care of the soil, and you build the soil. And obviously, my focus is on building the microbial community. But I mean, you could be putting other nutrients in the soil as well. But basically, you feed the microbes. That's the idea. You feed the microbes. Let the plant take the nutrients from the microbes. That would be the idea.
Yeah, and plus by default, the prey predator theory comes in. You have a tremendous amount of bacteria, and your protists are going to show up. They're going to come out of the woodwork and start doing that.
Yep.
Yeah, supply more building blocks.
Mm-hmm.
So his next question is, when taking a typical soil sample, the results are shown in chemical analysis. What biology is attached to that chemical result?
None.
None. None. As far as I know, none. We never have our agronomy. Much of our agronomy for years and years since I was a student has been devoid of biology. And, I haven't had agronomy lately, but I suspect it's still the agronomy that I had, very similar to the agronomy that I had. And it's very chemical oriented and soil physics and soil chemistry. And they do mention, there's a little section in the book, where it may mention something about microbes, mycorrhizae and their role, and soil microbes, but it's very minimal. It's very minimal amount included. So, and none in the soil test. It's under the paradigm that it's all about the chemistry, right?
Mm-hmm.
It's under that paradigm. And of course, that's easy for us. That's easier to look at chemistry because we can measure that real easy, and we can put chemicals on the soil. And we know it works. There may be negative outcomes, but we know it works. We could use chemistry, of course.
Do we know any types of specific biology that break down specific nutrients, like say calcium, boron, etc.? Have we narrowed it down to specific?
Yeah, you can get those microbes like that, and people are trying. When they try to make a consortia, right, for companies making biostimulants, right, they'll usually say, well, listen, we want a microbe that's a fixer, we want a phosphate solubilizer, we want something that's going to solubilize calcium, so something like that, right? So pick, put them together and then use them like that. Okay, so yes, it's at a level, but it's not as sophisticated as you might think. The 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 consortia people make up. Nothing wrong with that. It's just a logical way to make it up, right? One like this. That doesn't mean it's doing that really well in nature. It may not do that really well in nature. So we are on a kind of a, still kind of a, we're not highly sophisticated in creating consortia for plants.
No, I agree with that 100%. I mean, it's been shown over and over again about, in vivo versus in vitro grown biology, and the in vitro will work for a period of time, but you got to keep replacing it and replacing it because they don't have the tolerance to survive in the environment.
There's nothing like rebuilding a living soil, right? Rebuilding your regenerative soil. There's nothing like that. There's no substitute for that.
No.
I mean, yeah, plants need endophytes and they can lose their endophytes and they can be at a disadvantage. But if you have a poor soil, you can't grow much there.
Yeah.
You can't. If you have a dead soil, you can't grow much there.
Like Elaine says, dirt. You got dirt.
You got dirt. You got to bring it to life. Yeah.
Agreed. And let's face it, it's not the one, it's the family of them.
It is. It's not the one microbe. It's a whole community. Yeah, and the plant will get the ones it needs, right? It'll take the ones that it needs, really.
And the others will assist and be there as things change or evolve.
Yeah.
To support the other parts.
Yeah.
Of the community. So it is literally it takes a village, right? It's the best way
and there's no money in it. There's no money in it. So big companies can't make money doing regenerative agriculture. I mean, they can't.
They're trying. They're fucking trying.
It's hard to sell something. It's hard to sell it, but maybe they'll come up with something that's helpful to sell.
Yeah, they're definitely trying.
Yeah.
Okay, our next question is: any recommendations on how to measure plant hormones in making plant amendments?
Well, there leads you down the path of understanding fermentation, composting, whether it's static, turned, inoculations. That's that whole next level of farming, because your metabolites are going to come as a direct result of those interactions through, again, fermentation, composting, any type of not so mechanical, but biogeochemical reactions and interactions. That's the muncher. So the muncher takes 5 different components: heat, pH, agitation, aeration, and flow. There's a better word for it. But bottom line is that those 5 components together create the release or the restructuring of these metabolites, whether they're hormonal or whether they're like endophytic. So this is really cool, James. This is literally discoveries that was made within the last few days of this phenomenon called the ESS muncher system that we did a couple of different batches, and the first batch came out on the 13th, and it had like hundreds of flagellates. Like I would say probably 25 to 30 flagellates in every single view of the microscope. Just tremendous inoculant. The next batch that came out, or let's skip the middle one because the middle one was a combination of two different things, which I don't like doing. I like to do the pure and then look at them, right? So the last batch was all ciliates and maybe two flagellates, if I was lucky in a view. And so we obviously learned that, okay, this recipe is not going to work for us under this environment. But as of day 14, the flagellates had begun to reduce drastically, and the ciliates are starting to move in. So as it's going anoxic, they're moving out. But this is a tote that got zero oxygen for 14 days. And so I have to believe these are a facultative flagellate, not a true aerobic flagellate, because otherwise they would have vanished within 24 hours as the BOD got so high due to all of the biology. Because this thing is like, a bio bomb. You put it on the microscope and there's just shit flying everywhere. So is that actually a thing that there's these facultative flagellates? I mean, I've never seen anything like this.
I don't know. Are they able to grow after you take them? Have you pulled them out and looked at them?
I've not plated them or anything like that. But bottom line is that we definitely went through a increase over the first probably 5.
So there's no oxygen around. They increased.
No, it's a water. It's in a 250-gallon tote.
Well, don't forget that they don't have to be alone, right? I mean, they may be in there with some other microbes that are anaerobic, and so they may have some way to live on those other anaerobes. I mean, they are not. I mean, obviously, you have a ferment, right? So it's not just one thing; it's a lot of stuff. So the interaction there could be an interaction happening, causing microbes to survive that might not otherwise survive.
So maybe they're actually pulling the hydrogen and separating out.
Something like that. Yeah, they could be getting some other nutrients partially degraded that they're sharing and getting, right? So they don't have to break it all down aerobically, right? Because they can get nutrients from these other microbes that are doing anaerobic respiration.
Gotcha.
So it could be coming from something else in the community, because it is a complex community you're talking about.
Yeah.
And I really don't. I don't know the answer. I'm just saying that it might not be just as simple as are they facultatively anaerobic or right? You see what I'm saying? I mean, they may be getting nutrients symbiotically.
Well, again, this is why I love you so much, is you're not afraid to hypothesize about some of these phenomenons that I run into in the field. Because otherwise, I have no options. It's not like I could say this is left or this is right. I'm in the fucking gray the whole time.
Well, it is the whole. Just think. I mean, this is a different mentality, right? But it's the whole community that you're looking at. The ferment in that ferment. There's a lot going on in that ferment.
Mm-hmm.
And it's all about the biology in there and the interactions, the symbiosis, so the community has different properties than the individuals in that community, right?
Biodiversity
Has different properties when you look at the whole thing, different features, more resist. Like that soil is more resistant, right? It's resistant, and plants can survive there, and plants get nutrients, get lots of microbes there, and they share microbes. You take that all away, and you have just some, a substrate that doesn't have, in which plants can't have resistance, and they're not tough, and you don't get as much resilience. So
Yeah. So we're going to go to the last question here by William. What is the best way to mitigate high salinity in compost windrows from spent mushroom feedstock?
Oh boy.
Biology.
Well, yeah, but the problem is that you need a salt tolerant bacteria that can help break them off of the CEC sites. And let's face it, organic matter is just total CEC sites. So that's the problem with once that salt gets introduced into organic matter. Like typically in soil, it's relatively easy. You just go with a double positive like calcium to knock off the sodium off the cation exchange site. But in compost, now you'd have to throw so much calcium at it that you would take it all the way up to probably 10 or maybe even higher. And so now you're all alkaline. You shut down the process of degradation. It's the exact same thing when we're dealing with compost and lemons. As soon as you add those low acid or high acidic lemons into the compost process, it just shuts down. It just stops. It stops degrading. So that's a hell of a bind. I mean, I would probably recommend that, depending on how high the salt is, that you attack it with at least 2 prongs, which would be some calcium. We tried the BioAg's Calmino, which I thought was going to do wonderfully in knocking those ions off the cation exchange site, but it really had very little result. So then now I would say either you go with a natural farming. What is that calcium mix they make?
WCA, water soluble calcium.
WCA soluble calcium. I did have some good results with that when I was dealing with my garden here in Oxnard, because we live right on the ocean, so we get a lot of residual salts coming in, mist and the fog and stuff. I tripled the recipe. Like Chris Trump got pissed at me because I used 3 times what he recommends, and I go, but I'm not growing plants. I'm trying to fix soil. So I would suggest that to him, and I also suggest reaching out to Fish Brew to try to get the product called Trifecta, because they did have a lot of sodium tolerant bacteria in their mix, and those bacteria are more than capable of associating and knocking those sodium sites and cleaning the sodium sites so that another ion could attach, or just go quicklime. But you start throwing like hardcore chemistry, now you're really going to lock or launch the pH up to the alkaline side. But that's the best I can do. I don't know, James, you got anything better?
Well, I mean, I agree with everything you said. I mean, the problem is how much salt, right? How much salt is in there? How saline is it? And if it's not too bad, you could use biostimulant microbes. Just put them in there. But it comes down to this. It comes down to, if your microbe is inhibited, it's not going to do any good. It's not going to, because if the microbe is inhibited on the outside, the plants can't get it to have, increased stress tolerance, right? So you have to have a microbe that really is resistant to the salt. So really, this is where you could go in and you don't have $1 million to do a research program up front to figure out where your best bacterium is, right, for that salt tolerance that's going to be not inhibited by salt. But you find one, you could put it on, and then that would enable the plants to grow. But the problem is, you I don't know how much salt it is, and you do have to find, and if it's too high, it's going to inhibit. So there's no point to. Well, I mean, it might be worth trying a biostimulant microbe just to see if that works. That's the bottom line. Everything that Leighton said. I'm sorry, Leighton. Go ahead.
Yeah, no, it's all good. I mean, another thing maybe is like a biohack. So go collect some Phragmites from a salt marsh, or another plant that is known to be highly salty, and mix that in with your mushroom compost so that you are introducing, these in vivo or in vivo bacterium that could very well do something similar to what
You could do that with roots, for example, the Phragmites roots, or I would go with any woodland grass. You could get those roots and grind those and put them on your seeds and see it, even put them in furrow and see if you could get it to take.
Mm-hmm.
But
all right.
the key is, you're right. The key is getting a salt tolerant one. So Phragmites is probably got some that are salt tolerant.
Yeah, that's a badass plant.
Yeah. That's what we work on, Phragmites. That's a
Dude,
All right, James, before your wife gives us a trouble.
Thank you so much, gentlemen. Thank you so much. Y'all have a wonderful evening.
all right, thanks, James.
Thanks,
We'll
James.
talk again sometime in the next 6 to 8 months.
Sounds great. Bye.
All right, thanks, buddy. Peace.
Bye-bye. So, guys, we do have some more questions, but don't forget, we're going to run the contest again. So all you have to do is put in hashtag compost to be entered, put it in the live chat, and we'll do the draw when we're through the rest of these questions. So, Leighton, I could also separate the mushroom feedstock and water it down. And I know you were talking before about mushroom substrate and breaking it apart because there was a problem if you didn't.
Yeah, there's a liquid that forms inside the bags. I'm assuming he's dealing with a bagged mushroom substrate. If it's already been composted, so if it's like loose, that's a different animal. But if it's in a bag and you see the liquid in the bottom of the bag, that is actually a biocide, and a very, very powerful one that's leaked by the fungi so that there is no competition. They're very aggressive, these mushroom cultivators or cultivars, when it comes to being in a completely sterile environment. They will go to the ends of the earth to keep everybody out, so they have total control of their media. So if you have that liquid, then you've got to get rid of that first. We recommend taking the block, breaking it open, not powderizing it, just bust it open, let it dry out, because the spores will still be in there, and then add that to your composting process. As far as watering is concerned, if you're gonna do that, you need to make sure that your water is rainwater or structured at the very least, because otherwise it's really not going to have any value. You need a high DO in it, as well as you need it to be magnetic, so that it's going to be able to have the ability to knock those sodium crystals or ions off of the cation exchange. But it's worth a shot, definitely. Just put it out in the rain for a couple of days. See what happens. See if you've reduced the salt.
Perfect. Now, they also said they were thinking about adding EM in labs.
I don't think that the purple sulfur bacteria are going to be able to do that. They're more focused on creating sulfides. So EM, probably not. Bacillus, again, I'm not sure that that's the right microbe to scrub your cation exchange sites. I think you need a microbe that is way more focused on that, with the ability to, again, steal an electron from that sodium or share an electron. That's going to weaken the bond and make the bond more susceptible to breaking. So that's why I'm suggesting salt tolerant microbes, not necessarily plant growth microbes.
Yeah. Well, it's like what you did when they sprayed your yard, and you went and found or got your friend to send you plants from beside a field that had all those chemicals sprayed on it because it had the right biology. So it's basically finding a similar circumstance.
Exactly. Using, biohack, using nature, using things that in nature can survive in an environment that's very hostile, and then composting them or liquefying, turning them into dissolved organic matter.
Yep. And guys, it's all about the biology, because it's the biology that breaks down right at the molecular level and splits those cell structures apart. So it's no longer Roundup or salt. It's a different, or it's broken down just to raw components.
Right, broken to raw components or shared electrons and broken the bonds.
Yep.
Those are the two things. Good, great question. So
he said it's in bulk and it's still very active.
Okay, so in bulk, do you mean in like, is it in bags still, or is it in like a super sack with individual blocks?
I think it may be actually a tractor trailer coming in and just dumping it out. But that's my.
Yeah, but either way, like if it's not bagged and it's loose and broken into small pieces, that's one thing. If it's in a bag and it's got the white mycelial wrap around it, again, I was talking with Craig about that today. We're chasing down the succession of saprophytic fungi, because you have like these aspergillus, which are very, very early in succession, and then you have the saprobes, which are at the very other end of the spectrum as far as evolutionary talent. There's this whole range in the middle of like, all right, so which one comes first? We know pretty sure it's like the water molds, then you get into yeast, then you get into aspergillus, oomycetes, and then it keeps going up the scale until you get to the saprobes. But that original, that white coating that you see on the outside of a piece of mycelium or on the outside of a mushroom block, that is designed to prevent oxidation or oxidative stresses, as well as UV, because both of those things will kill the fungi. So that's protection coating. It's not the fungi itself, it's their excretion, it's their biofilm, the fungi's biofilm, to protect it from the environment that's not suitable for them to live in.
Well, it's like what the astronauts did on the space station, putting the biology on the outside in open space, and they got the coating to protect what was inside.
Yep, the biofilms.
Yep. Biofilm is so important in our bodies as well, because I don't know if you've ever followed Dr. Lee Layton, or Dr. Lee. So one of the things he talks about is the biofilm in our stomach, that if it's not there, the cancer cells are activated. If it's there, then the cancer cells are being able to be found by your natural biology and destroyed. But if it's not there, you've taken antibiotics, etc., it destroys that particular biofilm, and then people are getting cancer.
Yeah, I think it's called a
mucus layer.
Yeah, whatever. Like you said.
I call it biofilm because it's basically the same damn thing.
Yeah, exactly.
Yeah,
it's a real thick one.
Yeah, exactly. Or it's supposed to be anyway. So
right. If you're healthy, it is.
Yes.
Go ahead.
Did you have anything more to say on the last question before I go to the next one?
No, we're good.
Is adding foreign biology to soils necessary, or is it a good thing adding additional diversity?
So again, it's all about diversity. Look, the biology is either going to do one of two things. It's either going to consume the biology if it doesn't like it, or if it's out of balance. There's going to be, we call them the great corrector, right? There's always a great corrector in every environment. That's where prey-predator comes from. You have too much prey, predators are going to come in and knock that balance back down. Maybe even swing it the other way to the point where now the predators start to die. And as the predators die, then the prey comes back in again. So the great corrector will always be there. So you should never be concerned about adding diversity to your soil, because again, you're either adding a biostimulant, a food for the existing biology, or you're adding microbes that can enhance your existing biology through horizontal gene transfer, or what was that? Something symbiosis that we were just talking about, biosymbiosis.
Yeah.
I got to remember that word, biosymbiosis. So, or through biosymbiosis, where these bacteria are achieving a greater success level of transferring the information and fucking with the genetics. Did you hear him saying that?
Genetic potential, brother. I've been talking about it for 3.5 years. The only way to get true genetic potential is through biodiversity.
Yep, agreed 100%, man. Agreed 100%.
Yep. Well, it's the evolution of our planet.
And just so you know, sorry, Ken, I've been saying the same thing for freaking years. It's you. If you're not evolving, you're dying. You're going off the slippery slope. You're going the other way.
Yep.
You're going to be replaced. So, if you want to hit genetic potential, you have to have as much diversity as possible. Otherwise, you're gone, man. You're going to get replaced by the next level.
Yeah.
Someone who does have, or some plant that does have the diversity.
Mm-hmm. Well, you look at the beginning of biology on our planet, we didn't have plants that, interacted well with the biology that existed. So they had to, like the Venus flytrap, collect flies and other forms of protein. But as the planet progressed, now we want more fungally dominant species in our soils, but without that base level, like you say, the predator prey, without the base, you have no food source for the next generation of biology.
Right. Exactly right.
Now, also on that point, guys, we talk a bit about terroir here, and that development of your seeds from one season to the next is also not just giving the benefit for the best plant, but it's changing that plant to fit the environment you're growing in. That's part of the gene transfer.
Agreed. That was one of the smartest things that some of the early on cannabis growers, hippies up in Northern California did, was they would take seeds and they just throw it into a corner and just let it pollinate and break seed, pollinate and seed, year after year after year. And then they would have a genetic that was perfect for their environment. So basically teaching the plant a new, and this is where I always get confused because you have Appalachia and you have terroir. I think it's the Appalachia that is your environment and the terroir is your influence. But again, fact check me, please, because I don't want to get, piss anybody off. But there's you as a human interacting also change that environment through the things that you do, like adding manure, having animals, cultivating the soil, fertilizing the soil, changing the plant species. You are changing that, if I'm correct, Appalachia with your terroir. So that's what it was explained to me by Frenchie. And again, I might have him backwards. I've been known to be a little dyslexic on some of the shit.
No.
Oh, dude, what killed me for years was ecto and endo. So, ectomycorrhizae is, well, you would think that ecto, right? Echo, well, that's on the outside, right? And endo is on the inside, right? Mm-hmm. But it's that is the case. But that's what I had to do to remind myself, what is the difference between the two? Because endo sounds like it's on the outside, but in the reality, endo is on the inside, and echo, is on the outside. So anyway, I don't know where that came from.
Well, because a lot, and I have the same problems of remembering. So it's those little tricks to remember things. The association is really helpful to people.
All right, well, there you go, Silly Lil. Thanks for that appreciation.
Okay, what is she saying? An appreciation signifies a specific.
So
okay,
is a specific geographical area, and it's geographical should also include weather patterns, right? Because the geographic is also alluding to the fact that because of your geographical location, you are going to have a specific weather pattern, which was all great until shit started to really change a lot.
The weather patterns change,
right?
Yeah.
Just like permaculture. I used to knock heads with permaculturists. I'm like, this is all great, you guys, but we already know, like, everything is changing so fast now that you can't count on that plot of land to grow those trees at this speed to this degree before something else moves in and changes it. You know, another tree species comes in, or another plant species, or grass, or whatever happens as a result of the shifting of the weather patterns. So maybe we need 3 categories, though.
Yeah, no doubt. Well, I know, like, our weather here, we're getting warmer and warmer and warmer, and I'm able to grow different species now than I was, 5 or 10 years ago. Like, it's just when the wind patterns in the higher atmosphere change as our planet is shifting, everything's going to change. And until we're back at 90 degrees and everything settles down, we're not going to know what's going to happen from year to year.
No, and I think the biggest thing is the jet stream, both the aquatic jet stream as well as the atmospheric jet stream.
Yeah.
Those are the things that we could count on that would remain relatively
Stable
for a season or for an annual year. But those now, I mean, we get these huge swings in the atmospheric
flow
jet stream versus and now they're also really highly concerned about the aquatic jet stream that goes from the Gulf of Mexico up to Greenland, because the ice caps are melting so fast, they're cooling the water faster than it used to, and therefore slowing down that convection of the heat, the hot and the cold water. So
well, it's why England is so warm in the wintertime when they're actually in the same area as Canada is, where we get freezing temperatures. It's because of that aquatic jet stream that they get that moist air. If that shifts, it's going to be an interesting situation.
Yeah. Again, it's an interesting time to be alive. It's kind of horrifying if you look at the biogeopolitics and all that shit, or I should say geopolitics, versus also the influence of these weather patterns is a little horrifying. But on the science side, I'm having a ball having conversations with James about the latest understandings of some of these successful evolutionary strategies.
It's amazing. Like, that's why I told Lily, observe, don't absorb, because things are so crazy. Just watch, pop some popcorn. It's all changing. The whole energy on our planet is changing. It's an amazing thing to watch.
And I think that's why there's so much strife, is there's so much positive energy that's coming out.
Yeah.
It's causing all of that negative reactions. But now we're getting into spiritual woo.
I was actually supposed to do a show with Dennis and his sons a couple of weeks ago on energy, frequency, vibration. I was going to bring out some more seeds that I have wanted to sow.
Yeah, love it. You chase that boy down. That family is amazing, just amazing.
Yeah. So, guys, before we go any further, I've only got 3 people that have entered for the contest today. So, anybody else that wants to enter, we'll give it guys a couple more minutes here, and then we're going to make the draw.
So we have one more question, Star?
Yeah. Do you need a pH pin in your soil?
pH is regulated by biology. And that's what makes it so friggin' complicated, is you got pH and you got EH. Reduction. So everything is in motion. The only time it's really important to use a pH is when you're dealing with pure chemistry. Because in pure chemistry, if your pH is off, you're going to have Newt lock, or you're going to have shutdown. Either you're going to go anaerobic, in an acid environment, or you're going to also, on the alkaline side, you're just going to shut everything down. So that's why that neutral pH is so important. But again, that's just chemistry. When you're dealing with biogeochemistry, then you don't necessarily worry as much about the pH. And this goes back to some of the real early work I did when I was working under Lane, which was I took a rhizo sheath, which everyone knows what it is when you grab a plant and you pull it out of the ground, there's this big cake of soil around the root ball. That's a sheath. And so I took the sheath and I cut it a cross section and I sampled different sections and parts of that sheath. And I found ciliates, I found nematodes, I found everything. So there were anaerobic pockets within that sheath that was supposed to be all aerobic. And when I brought this to Elaine's attention, she kind of like, well, again, we had a great relationship. She would just like, Clayton, don't talk about that.
Nobody's supposed to know.
Anaerobes, no anaerobes. And I don't blame her because of all the shit she went through. So, again, it's, at least in my mind, it gave me the trust and understanding that we need anaerobes, we need facultatives, we need aerobes, we need all of them. We cannot live with only one group or just this part or just that section. It really has to be the full spectrum.
Well, and the thing is, most people still haven't grasped the concept that the biology literally changes the pH at the root hair itself to support the biology that's necessary. Like if it's a grain of sand that root hair is beside instead of something else, then it needs specific biology that mines that grain of sand. So
at that pH.
Exactly.
Right? That pH. They need to increase or decrease the pH so that organism has the ability to actually mine it. And that's usually through, recalcitrant or acid type of chemical, or what do we call cytoplasms that come out of excreted by the organism to do its work.
Mother Nature, she had all this figured out and we screwed it up. Now we're trying to figure out how to fix it. But I think the muncher juice is going to be one of the keys to being able to quickly change things back. And I'm interested in what is going to happen at the football field. And I'm glad you're able to track it to see how quickly the chemicals destroy what was built, and then hopefully get another opportunity to flip it around again and rebuild it and see how long it takes to rebuild it.
Well, I really appreciate Chris for allowing me to stay on as a consultant, even though, we basically got a bitch slapped by the egos of management. But either way, this is one that I'm going to tell him to watch, because I think James hit the nail on the head with that understanding that we had such a diverse, incredibly powerful biological system up and running. That's the only reason these plants aren't dead yet. Because Chris and I are just scratching our fucking heads going, look how much shit he's throwing on this field. His nitrogen's through the roof. His calcium's through the roof. This is way too much for these plants to take up. But he just keeps fucking pounding them. And for some reason, they're staying alive. They're not performing as well. We know that for sure. But they're somehow staying alive. So it has to be the endophytes and that symbiosis, the biosymbiosis that's happening.
Well, there's enough biology, there's enough workforce there to make it still happen. Just it's getting worse and worse and worse as the biology drops back. And then you're going to have to flip it around and get the biology back to take the components and literally use them up again and seal them away. It's like in Russia, where they drove across the radioactive area in the Ukraine, and the radiation came back that was stored in the fungal hyphae. But she's going to take that back, build more fungal hyphae, store those molecules again. It's crazy.
All right.
Well, guys. Oh, sorry. I was going to run the contest.
I was going to say, let's go ahead and run the contest. And as soon as this is done, I'm going to actually pop back to work. So this gives me a great opportunity to finish the day up.
Cool. Chris, you got 10 seconds. Oh, you already put in #compost. So, all right, guys, we're going to run the draw right now so Leighton can get back to work and make some more fantastic compost. And who we got here is Yellow Skunk Farms.
Sil, I thought you were going to get it.
She said if she did, that we'd have to redraw. But my friend, you just have to send me an email at it'sallaboutthebiology.com or at gmail.com, It's all about the biology at gmail.com. We have had an issue with the page, and that's being fixed on the website. So that should be fixed right away. But send me the email, and I'll get the gift out to you as soon as I can.
Ken, did that client of mine, Brandon's cousin or whoever, ever get in touch with
No.
you? All right,
No.
I'll text him a reminder.
Yep. Yeah, that would be much appreciated. And we got to get somebody on with the redox, Leighton. Somebody with redox.
Oh, dude, that's a fucking rabbit hole and a half.
Exactly. And I haven't went down it yet.
I went down pretty hard, but it's just so complex. I know a person that could do it. It's just a matter of right now they're just so busy with what they're doing that I don't believe they'd have the time to actually put because it's going to need to be more than a chop up chat. There's going to be some documents that are going to have to be shared because these graphs and stuff are going to help to explain it. Because if you just talk about it, forget it. You're never going to get it.
Yeah,
I mean, we went down that rabbit hole during COVID, and it was like I think probably 3 or 4 months of talking about it weekly before we really started to click on understanding the difference between these two radically different environments. And again, pH is the measurement of electrons, EH is the measurement of photons or protons.
Yep.
I'm sorry, protons. So it's now you're talking about subatomic particles. This is no light fucking science.
That's where I'm at, though, Leighton. It's all down at the molecular and the subatomic. It all fits together. But like Cake says, we need to do a series on it.
All right, I will plant the seed. I don't know how long it's going to take for that seed to actually germinate into something, but I will plant the seed.
And throw a seed at Chris Trump. We'd love to have see him on here too.
Oh, he's been blowing up lately, man. He came up with this IPMO. I don't know if you've been paying attention.
No, I haven't.
All right, so by using IMO in conjunction with bug parts, so a pest that you don't want, you collect a bunch of them and you add them or blend them into your IMO mixture.
Yeah.
And you put it out, and it basically grows specific biology. And again, I have to believe it's a yeast and not a saprobe, because you're not getting saprobes in those IMO collections. But either way, when you spray this as a liquid IPMO, when it hits the targeted species, it encapsulates them in a yeast. So
really
Shango just released a podcast on it, on shaping fire. So why don't you go to that one? I know he was also, I saw him doing a short on his site about it. And then he recently was on with Brian, Rubber Ducky Isopods, and somebody else, not Marco. And he did an interview. So like I've seen you pop up like 3 times in the last couple of weeks. So he's definitely found something very interesting. And Microbes with Marco has also done a couple reels on it as well. So there's some resources for you to check out.
Well, Nathan, who's our guest next Monday, says he can talk about LIMO when he comes on.
There you go. Well, this is not LIMO, but it's IPMO. So it's individual pest management organisms. So yes, you turn it into a liquid, but it's totally different than typical LIMO. It's an IPMO or liquid IPMO.
Cool.
Yeah, maybe you can check it out, Nathan, and dive down that rabbit hole and come back with some some interesting anecdotal information about what Chris is up to.
Cool. And like he says, I know IPMO, so there you go.
Okay, all right, so he does.
And he says it's actually LIMO.
Well, there's a difference. LIMO is liquid IMO.
Okay.
IPMO is a specific IMO that's targeting a specific pest. So Nathan, just to make sure you're actually 100% clear on that, but check it out. Go check out Chris's Trumps. Check out Shaping Fire. Check out Rubber Ducky Isopods podcast on it, and I'm sure you'll have a lot more information about it.
Cool. Leighton,
right.
your puppies wanted to go early and you got to go back to work. So guys, peace out in chat. Hopefully Andy Lopez is feeling better for this Wednesday and we can get that show done. If you guys didn't watch my interview with Gardner Scott, check that out from last Friday.
Nice.
And just by the way, Leighton, Scott is interested in having you on over the winter on his show.
Cool. Absolutely.
Yeah.
He can get a hold of me. He knows where I am. I'll hop on with him in a heartbeat.
You bet. Peace out, everybody.